Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Excel International A-Level and GCE Biology Practicals. This is core Practical 14. Investigate the effect of different antibiotics on bacteria. So I began with a theory. We know that antibiotics can affect the growth as well as the increase in the number of bacterial cells. So using antibiotics is going to decrease the bacterial growth or it's going to kill bacteria. We have two different types of antibiotics. Some are called bactericidal and others are called bacteriostatic. The bactericidal antibiotics will kill the bacteria, while the bacteriostatic antibiotics slow down the growth and reproduction of bacteria. We can observe the effects of antibiotics by observing the clear zones on the agar plates around the antibiotic filter paper. You also need to remember that for any culture, microorganisms require a good carbon source because, of course, carbon is going to be used in energy provision. They require a good nitrogen source as well as specific minerals for growth. So these should be included in the agar medium. During culture, you need to use aseptic techniques, which include ensure that all surfaces are disinfected. You could use an alcohol to wipe the whole surface. Also, you can use sterilized equipment. Ensure that all equipments are autoclaved, at least those that need to be disinfected. And then you could use a Bunsen burner flame to disinfect the work area. You also need to wash your hands before and after the experiment with soap. You need to carry out the experiment using the aseptic techniques to ensure that all surfaces are clean and your hands are clean. You remember you first pour the agar into the petri dish, the agar will solidify, and then on top of the agar, you will use a sterilized loop and then inoculate with microorganisms, and then using sterile forceps, you'll pick up the filter paper that has been previously soaked into the antibiotic and place it in specific areas. Remember, in this process, you have to make sure you minimize the exposure of the agar to the open air. So when you're placing the filter paper, open the petri dish lightly and carefully place the filter paper onto an inoculated petri dish. You immediately cover the petri dish. You also need to flame the forceps and then pour them down. Then place a small piece of tape on each side of the petri dish to hold in position. Then you will invert the petri dish and incubate at a temperature about 30 degrees. Do not inoculate at a temperature higher than that because in case these microorganisms are pathogenic to the humans, there is a chance that you could culture them at perfect conditions for them to grow and cause infection. So culture below 30 degrees and then for 24 hours. Then wash your hands and disinfect the work area. And later, observe the agar plate after 48 hours and measure the diameter of growth inhibition zones around the antibiotic soak disc. In this experiment, since the dependent variable is the diameter of the clear zone around the filter paper soaked in antibiotics, if you can measure that, then you have the results to know which sample has the greatest antibiotic effect. So this brings us to the end of Core Practical 14, which was investigating the effect of different antibiotics on bacteria. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.